The octopus paradox. Adaptive, but doomed. Molecular genius. Biological deadline. The animal that rewrites its own code. Built for brilliance, not survival. Octopus RNA editing. How octopuses rewrite messages in their cells and why they still die after reproduction. Octopus RNA editing is a biological process that lets an octopus alter certain genetic messages after they are copied from DNA, changing what proteins get built without changing the underlying genome. It matters because it offers a real example of fast, flexible molecular adaptation inside a single lifetime, especially in the nervous system, while also exposing a hard limit. Even a highly sophisticated animal can be locked into a life cycle that ends in programmed decline after reproduction. Here's the tension. RNA editing can make an octopus's neural machinery more adaptable, potentially helping it cope with shifts in temperature and environment. But that same animal still follows a reproductive script that drives a steep physical breakdown. In other words, one system supports flexibility and survival in the moment, while another system imposes a non-negotiable ending. By the end of this explainer, you'll understand what octopus RNA editing is, how it works at the molecular level, where it helps, what it cannot do, and why the octopus death spiral is not a trick for immortality but a hormone-driven program tied to reproduction. The story turns on whether rapid molecular flexibility can meaningfully extend survival when the organism is still governed by a built-in self-destruct sequence. Key points. Octopus RNA editing mostly refers to A to I editing, a chemical change in RNA that can alter the protein a cell builds. RNA editing is very common in the nervous tissue of many cephalopods, which suggests that it has something to do with how the brain works and how it can adapt. RNA editing can act like a temporary patch applied to genetic instructions, allowing proteins to be tuned without changing DNA. A well-studied example links RNA editing to temperature adaptation in neural ion channels, helping maintain nerve function across different thermal environments. This flexibility has costs. Heavy reliance on RNA editing can constrain long-term genome evolution and increase regulatory complexity. The post-reproduction decline in octopuses is not cheating death. It's a hormonally orchestrated cascade linked to the optic gland and reproductive state. Extending lifespan would likely require altering endocrine control of reproduction, not turning up RNA editing. The deeper lesson is that intelligence and molecular sophistication do not automatically produce longevity or technological trajectories. What it is, octopus RNA editing is the modification of RNA molecules after they are transcribed from DNA. DNA is the long-term genetic archive. RNA is a working copy used to build proteins. Editing changes the RNA letters in specific places, which can change the amino acid sequence of a protein and therefore change its behavior. The best-known form in octopuses and related cephalopods is adenosine to inosine editing, often shortened to A to I editing. In practice, the cell reads inosine as if it were guanosine. That single chemical swap can change the meaning of a codon and alter the protein that gets built. What makes octopuses stand out is not that they can edit RNA at all. Many animals can, but the apparent scale and functional emphasis, particularly in neural transcripts. In the cephalopod nervous system, editing can affect proteins involved in electrical signaling, synaptic function, and cellular transport. RNA editing differs from gene editing in the CRISPR sense. It does not rewrite the DNA blueprint. It does not permanently change inherited genetic information. And it is not a conscious process that the octopus decides to use like a tool. It's a biochemical pathway regulated by enzymes and cellular conditions. How it works. RNA editing begins after DNA is transcribed into a precursor RNA molecule. That RNA contains regions that may code for proteins, as well as regions that will be removed or processed before translation. A to I editing is carried out by enzymes called ADARs, which recognize double-stranded RNA structures. These structures often form when RNA folds back on itself or when complementary sequences pair. Once bound, an ADAR enzyme deaminates adenosine, converting it into inosine. Next comes interpretation. During translation, the ribosome reads the edited RNA. Because inosine pairs like guanosine, the codon can be read differently, and a different amino acid may be inserted into the growing protein chain. A small change at the right position can have outsized effects, especially in proteins that act like switches, channels, or motors. The cell must then deal with the results. Edited proteins must fold correctly, integrate into membranes if needed, and interact with other molecules. This procedure adds regulatory burden. The organism must ensure editing happens in the right tissues, at the right times, and at the right rates. Too little editing may reduce adaptability. Too much may disrupt stability. A helpful analogy, used carefully, is that DNA is the master recipe book, and RNA is the printed recipe card for today's cooking. RNA editing is like altering a few words on the card for a specific meal while leaving the cookbook unchanged. You get flexibility now, but poorly controlled edits can cause inconsistency. Numbers that matter. The most important number in Octopus RNA editing is not one universally agreed upon statistic, but the general pattern. Cephalopods can show unusually high levels of recoding edits in neural tissue compared with many other animals. In plain terms, more of the nervous system's protein instructions are subject to letter-level modification after transcription. A second practical number is the fraction of edits that are recoding versus non-coding. Many RNA edits in many species occur in non-coding regions and may influence regulation rather than changing protein sequence. What draws attention in cephalopods is the emphasis on edits that can directly change amino acids in proteins relevant to neural function. A third anchor is temperature sensitivity. For neural signaling, the meaningful thresholds are not abstract molecular counts, but functional stability across thermal environments. Ion channels and synaptic proteins can behave differently as temperature changes, affecting speed, timing, and reliability of nerve impulses. Editing that preserves function across cold and warm conditions can translate into real-world survival and hunting performance. A fourth is timescale. DNA mutations spread over generations. RNA editing operates on the timescale of RNA production and turnover, hours to days in many contexts, making it a mechanism for within-lifetime tuning rather than evolutionary change. A fifth is energetic and regulatory costs. Editing is not free. The organism must maintain enzymes, manage off-target risks, and coordinate editing with development and physiology. The more an organism depends on editing, the more it must protect RNA structures and sequences that enable the process, which can place constraints on how freely the underlying genome can drift. A sixth factor is lifespan, as it defines the limits. Many octopus species live relatively short lives, often on the order of 1 to a few years depending on species and environment. That short window shapes what adaptation can mean. Improving performance now may matter more than building long-term maintenance systems that support decades of survival. Where it works and where it breaks. RNA editing is well suited to problems where small molecular changes can stabilize performance in changing conditions. Neural systems are an obvious target because they rely on finely tuned proteins that sit on the edge of instability. Ion channels, receptors, and signaling proteins must operate quickly and reliably in environments that can shift. It also works when the organism faces variable conditions across geography or seasons. If temperature, salinity, or oxygen levels vary, a mechanism that adjusts protein function without waiting for generational turnover is valuable. Where it breaks is in problems that require irreversible structural change or multi-system redesign. RNA editing does not rebuild organs. It does not create new limbs, new cell types, or new metabolic pathways from scratch. It tunes what already exists. It also serves as a universal explanation for intelligence. RNA editing may support neural flexibility, but intelligence is a multi-layered phenomenon involving brain architecture, learning capacity, sensory integration, and behavior. Editing is one contributor, not a single master key, and it breaks completely as a route to immortality. The post-reproduction decline in octopuses is a major coordinated response, not fixable by tuning a few proteins. It is a coordinated endocrine and metabolic shift. If you want to change that trajectory, You have to intervene at the level of hormonal control and life history strategy, not just RNA message editing. Analysis, scientific and engineering reality. Under the hood, Octopus RNA editing is a chemical recoding of RNA bases that can change proteins at specific positions. The engineering advantage is modularity. Instead of hardwiring every protein variant into DNA, the organism can produce a baseline transcript and then apply conditional edits to generate functionally distinct versions. For claims about adaptive value to hold, three things must be true. First, the edits must be targeted to functionally important sites rather than being mostly noise. Second, edited proteins must be expressed at meaningful levels in the relevant cells. Third, the editing patterns must correlate with environmental or developmental conditions in a way that plausibly affects survival or performance. What would weaken the interpretation is evidence that most edits are neutral or harmful baggage, or that edited transcripts rarely get translated into proteins that matter in the living animal. Another weakening factor would be if editing patterns are highly inconsistent and not reproducibly linked to specific conditions. One common confusion is equating molecular novelty with organism-level control. Even if an octopus can generate many protein variants through editing, that does not mean it chooses them in a strategic sense. The regulation is biochemical. The intelligence is behavioral. They can interact, but they are not the same thing. Economic and market impact. In biomedical and biotech contexts, Octopus RNA editing is mainly important as a model. It shows that natural systems can safely change RNA on a large scale in complicated nervous tissue, which is important for treatments that want to edit RNA temporarily instead of making permanent changes to DNA. If RNA editing-based therapies work broadly, the beneficiaries would include patients for whom reversible, dosage-tunable interventions are preferable to permanent genome edits. It also matters for neurobiology and protein engineering. Edited protein variants can reveal which amino acid substitutions preserve function under stress. For practical adoption in medicine, the limiting factors are not octopus biology itself, but delivery, specificity, and safety. Getting an RNA editing tool to the right cells at the right dose with minimal off-target effects is the central challenge. Total cost of ownership shows up as manufacturing complexity, clinical monitoring, repeat dosing, and long-term safety surveillance. Near-term pathways look like narrow, well-defined clinical targets where transient editing is enough. Long-term pathways would involve broader tissue coverage, better programmable specificity, and robust control over when and where editing occurs. Security, privacy, and misuse risks. The most realistic risk is misunderstanding and overclaiming. Octopuses edit RNA can be misread as octopuses rewrite their genes at will, which fuels bad science communication and misinformation. In a technology context, the plausible misuse vector is not octopus secrets but the broader idea of RNA editing as a platform. Any editing technology has dual-use potential if it can be redirected toward harmful ends. That is why standards, auditing, and safety testing matter, especially for tools that could theoretically affect nervous tissue. A subtler risk is over-optimism about reversibility. RNA editing is transient at the molecule level, but biological systems can have lasting downstream effects. Changing a signaling protein during a critical window could produce long-lasting changes in development or neural circuitry. Not permanent does not automatically mean low consequence. Social and cultural impact. Octopuses reshape public intuitions about intelligence, agency, and what counts as a mind. Their nervous system is distributed, their behavior is flexible, and their life history is radically different from that of mammals. That combination pushes culture away from a human-centered template. In education and public understanding, Octopus RNA editing offers a concrete way to teach the difference between genotype and gene expression, between DNA and RNA, and between permanent and transient biological change. It also nudges people toward a more realistic view of adaptation, not just slow mutation, but also dynamic regulation. Second-order impacts include ethics. As people learn more about cephalopod cognition, questions about welfare standards, research practices, and food systems intensify. Better understanding of biology can change how societies justify treatment of certain animals. What most coverage misses. Most coverage treats RNA editing as a superpower and the death spiral as a tragedy. But the more revealing point is that these two features may be compatible parts of a coherent life history strategy. An organism can be optimized for short, intense performance and rapid reproduction, rather than long-term maintenance. In this view, having the ability to change molecules helps with survival and hunting for a short time as an adult, while a planned decline after reproduction helps protect the... Another overlooked element is that flexibility has a governance cost. If you expand the space of possible protein variants, you also expand the space of possible failures. That forces the organism to evolve robust regulatory control. The cleverness is not just the editing, but the system that keeps it from becoming chaos. Finally, the death spiral is often described as self-starvation, but mechanistically it looks more like a coordinated metabolic and hormonal reprogramming. That matters because it shifts the question from moral storytelling to biology. What signals flip the switch? What pathways execute the decline? And what is the evolutionary advantage of making that switch so hard to reverse? Why this matters. Octopus RNA editing matters to three groups in different ways. For biology, it expands the catalogue of how complex nervous systems can be regulated and adapted. It suggests that high-level RNA recoding can coexist with sophisticated behavior, and it invites new hypotheses about neural resilience under environmental stress. For medicine and biotechnology, it strengthens the case for RNA-level interventions as a serious design space. If nature can run extensive RNA recoding in a complex animal, it motivates the search for safe, controllable, reversible editing strategies in humans. For society, it reframes debates about intelligence and welfare. If an animal can solve problems, learn and adapt, then the ethics of how it is handled in labs, fisheries, and aquaculture become harder to ignore. Important things to pay attention to are not celebrity news, but scientific developments. Better understanding of which genetic changes lead to noticeable protein differences in living tissue, more straightforward experiments connecting these changes to behavior and survival, and improved knowledge of how the optic gland affects hormone changes after reproduction. Real-world impact. A neuroscience lab looking at how brains stay stable under stress can use cephalopod RNA editing as a guide to find adjustable protein sites in ion channels and synaptic machinery. A biotech team building RNA editing therapeutics can look at the octopus as proof that large-scale RNA recoding can exist in nature while still learning humility from the regulatory burden it implies. A marine conservation program can point to the same biology, to argue that these are not simple animals and deserve handling standards that reflect their cognitive complexity. A science educator can use octopus RNA editing to teach a modern, accurate view of genetics. DNA is not destiny in a simplistic sense, because RNA and protein regulation shape what genes actually do in real time. Frequently asked questions, SEO-driven human answers. Can octopuses edit their RNA? Yes. Octopuses can chemically modify RNA molecules after they are transcribed from DNA. The best-known mechanism is R-to-I RNA editing, which can change how a codon is read and therefore change the protein produced. This procedure does not rewrite DNA. It changes the working copy used to build proteins, making it a flexible, reversible layer of genetic regulation. Is octopus RNA editing the same as CRISPR gene editing? No. CRISPR typically targets DNA to make edits that can be permanent and heritable if they affect reproductive cells. Octopus RNA editing changes RNA, which is transient. You can think of it as modifying messages rather than rewriting the archive. Why is RNA editing vital in the octopus nervous system? The nervous system depends on proteins that control electrical signaling and synaptic timing. Those proteins can be sensitive to environmental conditions like temperature. Editing can tune key sites in those proteins, potentially helping maintain stable nerve function when conditions change. Does RNA editing explain octopus intelligence? It may contribute, but it does not explain intelligence on its own. Intelligence emerges from brain organization, learning, sensory processing and behavior. RNA editing is one molecular tool that can support neural flexibility, but it is not a complete story. Do octopuses sacrifice their bodies to evade death? No. After reproduction, many octopus species undergo a hormonally driven decline that leads to death. It is better described as a programmed life history pattern than a strategy to evade death. The animal is not transferring consciousness or regenerating into a new self. It is completing a reproductive cycle that ends in systemic breakdown. What causes the octopus death spiral after laying eggs? The best supported picture involves endocrine signaling linked to the optic gland and major shifts in metabolism and hormone production after reproduction. The behavior looks like a coordinated cascade: altered feeding, maternal guarding, tissue decline, and eventual death. Could changing RNA editing make octopuses live longer? It is unlikely that simply increasing or decreasing RNA editing would override the reproductive decline program. Lifespan extension would more plausibly involve endocrine control of reproduction and senescence pathways. RNA editing may influence resilience and performance, but the point of no return appears tied to hormonal state. What can humans learn from octopus RNA editing? It offers a natural example of extensive, regulated RNA recoding in a complex nervous system. That informs basic biology and motivates therapeutic ideas that aim for reversible edits. It also teaches a boundary lesson. Molecular flexibility does not automatically produce longevity, social complexity, or cumulative culture. The road ahead. Octopus RNA editing is best understood as a powerful, carefully regulated way to adjust protein function without rewriting DNA. It can support adaptability in the nervous system, especially when the environment challenges neural stability. But it does not grant biological escape velocity from the broader life history constraints that evolution has built into the animal. One scenario is that future research ties specific editing patterns to concrete behavioral advantages under changing conditions. If we find clear connections between changes at specific sites and noticeable performance results, it could help us create a better model of how RNA recoding affects actual behavior. A second scenario is that the field finds limits. Editing may be widespread, but only a smaller subset of edits meaningfully changes proteins in living tissue. If we see that most edits are buffered or rarely translated, it could lead to a more conservative interpretation focused on a smaller set of high-impact sites. A third scenario is that octopus biology becomes a design inspiration for human therapeutics. If we find that programmable RNA editing tools work very accurately and safely in medical use, it could result in a wider move towards treatments that can be reversed and controlled by dosage. A fourth scenario is that the reproductive decline mechanism becomes a clearer, targetable endocrine map. If we see precise identification of the hormone pathways that flip maternal behavior into systemic breakdown, it could lead to deeper insights into how evolution programs aging and senescence across species.